Different gland in our body. Example, hypothalamus present at the base of our skull, pituitary gland below the hypothalamus, and pineal gland, third ventricle of brain, thyroid inferior to the right, parathyroid backside of thyroid gland, thymus gland present at
functioning of hypothalamus. Hypothalamus contains osmotic receptors that react to the concentration of sodium, potassium, carbon dioxide, and chloride. When the blood pressure is very low, these osmotic sensors and baroreceptors intimate the kidney to release or store water and maintain the concentration of these substances. Hypothalamus maintains the internal body balance, that is homeostasis. It connects endocrine system and nervous system. It is involved in many body functions, that is childbirth, sleep cycle, maintaining body fluids, etc. That's
I am Vishwanath Mandal from Group A, and our group members are Udar, Asta, Rishabh, and Swati from roll number 1 to 6. And today we are here to present a seminar on endocrine system, pituitary glands. Endocrine system is a messenger system in an organism which comprises of feedback loop of hormones that are directly released into the circulatory system and that target and regulate distinct organs. There are 8 endocrine glands: hypothalamus gland, pituitary gland, thyroid gland, thymus gland, pancreas, adrenal gland, and gonads. Pituitary gland. Pituitary gland is called the master gland because it secretes several hormones that control other adrenal endocrine glands. It is a T-shaped structure about 1 to 1.5 centimeter in diameter. It lies in hypocele fossa in the sella calcica of the sphenoid bone. It attaches the hypothalamus by a stalk called infundibulum. The pituitary gland secretes several hormones. These hormones play an important role in the regulation of growth, development, metabolism, and
Pituitary gland is divided into 3 parts: anterior lobe, posterior lobe, and pars intermedia. Anterior lobe is also known as adenohypophysis. Anterior lobe is composed of epithelial tissue. It is further divided into 2 parts: pars distalis, which is a larger portion, and pars tubralis, which forms a sheath around the fundibulum. And posterior lobe is also known as neurohypophysis. It is composed of neural tissue, and posterior lobe is also divided into 2 parts: infundibulum and the pars nervosa. And the third region is pars intermedia, which itself migrates and disperses in the adjacent parts of the anterior lobe. Types of anterior cells, anterior pituitary cells and their hormones. There are 5 types of cells present in the anterior pituitary, and it secretes 7 different types of hormones. First one is somatotroph, secretes growth hormone, which is known as somatotropin, and it helps in growth development and and thyrotroph secretes thyroid stimulating hormone, which is also known as thyrotropin, which is also known as thyrotropin, and it stimulates and regulates and controls the secretion of thyroid gland. Gonadotroph secretes 2 gonadotropin, follicle stimulating hormone,
Formation, storage and release of thyroid hormone. The process of synthesis and the secretion of the thyroid hormone occurs in 8 steps. First, iodine trapping. The process starts with the trapping of iodine by actively transporting it from the blood to the cytosol. Second, synthesis of the thyroglobulin. By trapping of the iodide trapping, the follicular cells synthesizes the thyroglobulin, which is the glycoprotein. It passes through the lumen of the follicle. Third, oxidation of the iodide. The iodide oxidizes into the iodine, and the iodine reacts with tyrosine. The process occurs in the colloid space called colloid space of the thyroid follicle. Fourth, iodination of the tyrosine. The iodine reacts with the tyrosine to form monoiodotyrosine T1 and diiodotyrosine T2. Fifth, coupling of T1 and T2. The T1 and T2 join together to form the thyroxine and triiodotyrosine. The two T2 join together to form thyroxine, and one T1 and one T2 join together to form triiodotyrosine T3. Sixth is the pinocytosis and the digestion of the colloid. The colloid re-enters into the follicular cells by pinocytosis and merge with the lysosome. The digestive enzyme in the lysosome break down the TGP cleaving of T3 and T4. Secretion of thyroid hormone, because the T1 and T3 and T4 are the lipid soluble, so it can pass through the plasma membrane into the interstitial fluid. Last is the transport in the blood. The T3 and T4 release into the bloodstream where it requires the transport protein called the thyroxine binding globulin. Now I will pass it over to Rohit.
Action of thyroid hormone. They stimulate synthesis of additional Na plus and K plus ATPase, which uses large amount of ATP to continually eject sodium ions from cytosol to extracellular fluid and potassium ions from extracellular fluid into cytosol. Next, they increase the concentration of enzymes involved in the cellular respiration, which increase the breakdown of organic fuel and ATP production. Third, regulates development and growth of nervous tissue and bones. Thyroid hormones are necessary for the development of the nervous system. It also required for the growth of the skeletal system, synthesis of many bones protein, and secretion of the growth hormone. Next is of the TRH. Okay, and then the B. TRH enters the hypothesial portal system and flows to the anterior pituitary, where it splits to secrete TSH. TSH here it carries the portal veins to anterior pituitary, stimulates release of TSH by thyroid. Third, TSH release into the blood stimulates thyroid follicles to secretion and growth of the follicular cell. D. In fourth, the T3 and T4 release into the blood by follicular cell. And fifth, on an elevated level of T3 inhibits the release of TSH and TRH and TSH, which is the negative feedback. Thank you.
Location, it is located on the dorsum, and it has four modular structure, and microscopically the posterior, thyroid gland
Good afternoon, everyone. We are the participants of Group A and Group number 4, and it consists of from roll number 16 to 20. The first is Rahul Kumar, then me, that it is Sorish Nanda, then Deepak Anand, then Habiba, and then Chandan. So our slide will start from the content. So the contents, our topic is pancreas. Then comes the control of secretion of glucagon and insulin. Then comes the insulin synthesis. Then insulin secretion is further divided into two parts, that is the glucose metabolism pathway and the incretin receptor pathway, and finally the conclusion and the acknowledgement. So I will hand over it to Rahul Kumar.
Pancreas. Pancreas is about 12.5 to 50 centimeter in length. It consists of head, neck, body, and tail. Exocrine cells are also called pancreatic and secretion enzyme. Endocrine cells are also called acid of Langerhans, pancreatic acid. Each pancreatic acid includes 4 types of hormones: proliferation, alpha cell, beta cell, delta cell, and alpha cell. Including glucose, beta cell, secretion insulin, convert glucose into glucose, delta cell, secretion, chromatostatic, secretion of insulin and glucose, fat cell, secrete pancreatic polypeptide, thyroid, chromatostatic secretion.
My topic is control of secretion of glucagon and insulin. So basically, it is a complete cycle in which how the blood glucose levels will be regulated in our body. So if there is hypoglycemic condition, if there is low glucose level, then glucagon will be stimulated from the alpha cells of the islets of Langerhans, which will increase the blood glucose level. Then when the blood glucose level increases to normal level, then it is okay. But if continues to increase, then hyperglycemic condition will take place. Then this hyperglycemic condition will inhibit the secretion of glucagon. Next comes the insulin. If the hyperglycemic condition continues to go on, then in that case, insulin will be released from the beta cells of the islets of Langerhans. This will lower the blood glucose level, and similarly the hypoglycemic condition will take place, and the cycle process is dependent upon glucose. So when glucose entry takes place from the glucose transporter, these glucose transporters are present in the pancreatic islets or in the beta cells or in the liver. Then when glucose entry takes place into the cell, then in the presence of glucokinase. Glucokinase means liver. This mechanism is taking place in liver. So the glycolytic pathway will start, in which pyruvate will be formed, and that pyruvate will enter into the citric acid cycle, in which ATP formation takes. As you can see in this ATP sensitive potassium ion channels, ADPs, first of all, ADPs sitting in this channel. But when ATP is synthesized, this ATP binds to this ion channel, and therefore it inhibits the potassium ion channels. Because of which the efflux of potassium is inhibited, because the potassium ion concentration inside the cell is more compared to outside. Then if the inside of the membrane is less polarized, then depolarization takes place. Inside of the cell is becoming positive. Then as a result, calcium ion channels open, which leads to the insulin secretion into the bloodstream. And this was the overall.
And incretin receptor. Incretin GLP and GIP protein hormones that regulate insulin secretion and release from the guts after eating. After GIP. Glucagon dependent insulinotropic peptide and GLP. Glucagon like peptides induce insulin release from the beta cell of pancreatic islet. And after that, as we know that water soluble protein, having G protein can activate adenyl silase, and adenyl Silase converts ATP into cyclic adenosine monophosphate, and cyclic adenosine monophosphate activate protein kinase, and protein kinase helps in the release of insulin. And that is the overall process. And conclusion, insulin secretion regulates, and insulin secretion is triggered by the elevated blood glucose level, particularly after meal. And insufficient insulin secretion.
We would like to express our sincere gratitude to Dr. Jyotirmay Sahu, who is the Dean of Head of School of Health and Allied Science. And we would also like to thank all the participants in our group for contributing in this seminar. And last but not the least, we would like to thank God because without His blessings, we would not have be able to complete this seminar. Thank you.